the problem set that you've had in your Canvas container. And I want to go over not just the answers, but how we work through these Mendelian inheritance problems. And this is important for a number of reasons, including the fact that they'll show up, questions like this will show up in the future on tests and quizzes. And that reminds me to remind you that we have a quiz on Wednesday, the 14th, day after tomorrow. And that quiz will cover cell division, mitosis and meiosis, and Mendelian inheritance. Okay, I'm going to share my screen with you. Do you see the worksheet?
Yes.
Okay. There are a number of questions here. I'm going to try to focus on those that illustrate particular points. So let's look at number 1 first. That's pretty straightforward. In guinea pigs, black fur is dominant over white fur. Now, how could an animal breeder test whether a black guinea pig is homozygous or heterozygous? Now, it's important for each of these questions to read them very carefully, look at every word, and derive as much information as you can from the various words that are used. So this second sentence here, how could an animal breeder test zygosity? What does that immediately tell you? And we talked about testing zygosity in lecture. Anybody have an idea? Well, it's a test cross. So we have a known and we have an unknown. So how do we know what the unknown is, or what the known is? We know that black fur is dominant over white fur. And so that means that the animal with white fur is homozygous recessive. And that means that the one with black fur could be homozygous dominant, or it could be heterozygous. So what do? We make a cross. And this is called a test cross. Now, if the unknown is homozygous dominant, big B, then we get the Punnett square here on the left. Big B crossed with little b. And you can see that what that gives us is all heterozygotes, which means they're all black. Now, on the other hand, if the unknown is itself a heterozygote, then we get the Punnett square on the right, where if you match up the gametes, big B, little b, two of them are heterozygotes and the other two are homozygous recessive, which means that half of the progeny will be black and half will be white. So if the recessive allele shows up in the progeny, then you know the unknown is heterozygous. If all the progeny are the same color, same as in this case the black one, then it was homozygous dominant. So is that clear? First of all, remember what a test cross is, because that will likely come up again. And secondly, how you work through the gametes to get an answer. Okay, I want to go then to the next question, which is important because it's a dihybrid cross. So let's read it. A short stem yellow pod pea plant is crossed with a true breeding long stem green pod pea plant. Now, the words true breeding here are important because what that means is true breeding is the same as an inbred. True breeding means that this phenotype has been selfed, had inbreeding generation after generation until it's homozygous at all loci. So a true breeding plant is a homozygous plant. So the true breeding long stem green pod, we can assign, well, let's read the rest of the question. Assuming that long stems are dominant to short stems. Okay, dominant long stems, and green is dominant to yellow. So the question asks, what are the genotypes and phenotypes of the F1 and the genotypes and phenotypes of an F1 times F1 cross, which by definition, that is how you get to an F2. Okay, so we can assign big L to long stem because we know that that's dominant. And that means that short stem is small case l. We can assign a big G to green pods because we know that green pods are dominant. That means that yellow pods are little g. And so the long stem plant then is genotypically big L, big L. The short stem plant is recessive little l, green pod, big G, yellow pod, little g. So we're going to make a parental cross, which means that we take the parent with dominant alleles, big LL, big G, times little LL, little GG. Could you mute your microphone? Thank you. So the big LL, big GG parent, the homozygous dominant one, will make two kinds of gametes, big L and big G, whereas the other one will make two kinds of gametes, little l and little g. So in answer to the first question, the F1 will be 100% heterozygotes. And the phenotype is long stems and green pods. So then we're going to cross one of these with another just like it. Take two F1s and cross them with each other. So what are the gametes? There's the so-called FOIL method that some people like to use, which means firsts, outsides, insides, and lasts. And what that gives you, this is a method from mathematics. The first is big L, big G, so we have big L, big G. The outsides are big L, little g, that's next. The insides are little l, big G, that's this. And the lasts are little l, little g. So these are the gametes that these F1 plants can produce. And so we make a Punnett square, listing the gametes on each axis. Across the top, big L, big G, big L, little g, and so on. Same thing on the other axis. And then we can fill it in. And I'm not going to go through all of these, but you can see what happens. Big L, so you have big L, big G, and so on. And so we can list the genotypes, which is what one of the things the question asked. Genotypes and phenotypes for the F2, the F1 by F1 cross. So if you go through this table, the Punnett square, you can see what the genotypes are and how many of each there are. And if we look at the phenotype in each of these squares, you can see that this one is long green. And if you count just the long greens, there are 4 at the top, 3 on the side, that's 7. Another long green, a long green, and a long green. Excuse me, just 2 long greens. So 7 and 2 is 9. So phenotypically, there are 9 long greens. And then we can count the long yellows. You can see there are 3. The short greens, there are 3. And the short yellows, there are only 1. There is only 1. So that is an illustration of how a dihybrid cross is done. Any questions about how you do a simple dihybrid cross? Okay, then I'd like to go to the next question. And this is an interesting one because it has to do with sex linkage to the X chromosome. In humans, hemophilia, known as bleeder's disease, is a recessive X-linked trait. Use a Punnett square to show the possible genotypes of offspring of a man who is a hemophiliac and a normal but heterozygous woman. So an X contains a normal clotting gene, and X star represents the gene for hemophilia. Okay, so we know that the man is a hemophiliac. That means that on his X chromosome, he's got a star. So he has a mutant X chromosome, and his other sex chromosome is a Y. The female gametes, female is a normal, but she's heterozygous. Okay, so she's X star. Her gametes can be either X or X star. And it asks us to make a Punnett square to show the possible genotypes. And you have that here, where you have the male on the left and the female on top. So we have X star X, we have X star XY, and X star Y. So X star is a normal female, not really normal because we should call her a carrier. She's got the mutation, but she doesn't have the disease because she has an X without the mutation. X star is again a female, has the disease because she has the mutation on each of her X's. XY would be a normal male. X star Y is a male with a disease. Why is that? Because he only has one X, and that X is mutant. Any questions about that?
So, Professor, there's no way that the male can be a carrier. He's either going to have it or normal. There's no carrier male.
That's correct. And that's an important point, a distinction between a carrier and someone with the trait. A carrier is not sick. Carrier just has the mutation, but is showing no symptoms. And in this case, as you said, the male, since he's only got one X chromosome and it's mutant, then he will be sick. Any other questions? Okay, I want to do number 4. Lesch-Nyhan syndrome is a metabolic defect caused by a lack of this enzyme, HDPRT. This disease is a result of an X-linked, so it's on the X chromosome, and it's recessive, and it results in mental retardation, self-mutilation, and early death. Now, a normal man marries a woman who had a brother who died of this disease. You are a genetic counselor who will advise them on the likelihood of their baby having the disease. So what are the possible genotypes for the woman? And what is the man's genotype? So what do we know about the woman?
The woman has a brother who had the disease.
Yes, that's right.
So that would mean she is either doesn't have it or she has one of her X chromosomes that has the recessive gene, but so she's just a carrier.
That's right. Yeah, we know that the woman and her brother, they had the same parents. So if the brother got the mutation, his sister may or may not have it because the brother is XY. And so if he gets a mutation, he's going to have the disease, and he died. But that means that with the same parents, the woman, since she gets X's only, she can either have a diseased X or not. And what do we know about the husband? Well, he's normal. Yeah? Say it again?
I was just saying he was normal.
Okay. All right, yeah, the husband is normal. And so we have two possibilities then. You're the genetic counselor, can set up two Punnett squares where you know that the husband is XY and the wife could be XX. And if that's the case, you're going to get two normal females and two normal males. There'll be no mutations if the wife does not have the mutation. However, if she does, you have a normal female XX, a normal male XY, a normal female XX star, and a sick male. And again, I should say this normal female is not really normal. She would be considered a carrier. So what are the chances then? You're telling your clients, you're giving them advice. What are you going to tell them about their chances of having a sick child? Just based on possibility 2.
So if they have a female, there's no chance that she has it, but if she has a male, it's only 1/4 no chance.
Yeah, that's right. Punnett square number 2 shows that females would not be sick, but half the males would. The chances are that they would be. These are all probabilities.
Just a quick question. There is a way to determine like for sure if she's carrying, right? Like the gene, like, you know what I mean? Right?
You
Like
mean
if she's the carrier of
in
the
advance, like if they were trying to decide whether to have a baby?
right.
Well, certainly with gene sequencing, and since we have the complete sequence of the human genome, yes, it should be possible to know in advance if there is a mutation on the woman's X.
Okay.
Okay, I'm going to skip over a couple here. Let's see. Oh, no, I want to do the next one because it illustrates another point, and that is a backcross. What we have been focusing on when we make a parental cross and we get an F1, and we've been going to the F2. And that means what? How do you get an F2? Anybody?
Katrina Vibbard. Isn't that when you cross an F1 with an F1?
Yeah, and that's what we've been doing in class and here when we take the parental cross, get a progeny, and we cross F1 times F1. Now in this question, we're doing something different. It's a back cross. So we take an F1 progeny and back cross it to either one parent or the other parent. And we ask, what do we get? So red fruit color is dominant to yellow. And we have a tomato plant that's homozygous for red crossed to one homozygous for yellow. What is the phenotype and the genotype of, first of all, the F1, and then the offspring of a cross of a back cross to the red parent and the back cross to the yellow parent. So we know that red is homozygous and red is dominant. So the red parent is big R, big R. The yellow parent is little r. And the F1 is all heterozygotes, all red. So now if we cross, and we've got two Punnett squares down here, but if we cross the red parent, back cross an F1, which is a heterozygote, to the red parent, that's shown here, and what you get is all red, because they're all either homozygous dominant or heterozygotes. However, if we back cross to the yellow parent, we're now taking a heterozygote F1, big R, times r, and that's shown on the right. And you can see, if you look at what happens in each square, that 50% of them are red. So in contrast to what you might see, the segregation that you might see if you did a self or a true F2 generation, if you do back crosses, you see something different.
Professor, I have a question.
Yeah.
Is that a possibility that it can be like one is 60% and another one is 40%, or it have to be half and half?
Well, look at the Punnett square and see what the possibilities are. I mean, what you see here are the gametes. And these are the only gametes that are possible. So what you see in the Punnett square are the only possibilities. In other words, you would not see, I mean, 50% is statistical, but you wouldn't see any other ratio besides that.
Oh, okay.
Okay. I want to make sure we get to number 9. because this, it makes yet another point. Again, this is a point we talked about in lecture, but it has to do with incomplete dominance. What if dominance is not complete? What do you see genetically? So this involves chickens. The farmer breeds a chicken with all white plumage, the chicken with all black plumage. Plumage is feathers. And all the F1 have bluish gray plumage. And if the two of the bluish gray birds are crossed, black, white, and bluish gray offspring are produced. So what is the mechanism of inheritance and the genotypes of the parents and the F1s and the F2s? Well, we'll choose black as dominant. And so that means that a black chicken is big B, or big B, little b. Could be homozygous dominant, or it could be heterozygous. That makes white recessive. That makes it little b, little b. Now the F1, we know from making this cross, we know that the genotypically, they're all alike. They're all heterozygotes and they're all gray. So both parents were homozygous. And so if we go to the F2, where we're crossing a big B, little b with a big B, little b, there's one big B, two big B, little b's, and one little b, little b. So only the big B is black. The two heterozygotes are gray, and the homozygous recessive is white. So to answer the question, what is the mechanism of inheritance? It's incomplete dominance. Incomplete because of what? Why do we call it incomplete?
Robert Johnson,
Uh-huh.
because it shows like it's like a mixture, right?
Yeah, a mixture, or some people would say a blend.
Blend.
It's unlike either parent, but like you say, a mixture of both parents. Not fully black, not fully white, it's in between. So remember that's the definition of incomplete dominance, that the heterozygote doesn't look like either parent. Okay, so that's the last one. Let's go to this one on the fly, Drosophila. The allele for dumpy wings is little d. It's recessive to the normal long wing allele, which is big D. The allele for white eyes is recessive, and that's called little r. And the allele for red eyes is dominant, that's big R. So you make a cross of a homozygous dominant, big D, big R, with big D, little r, little r. And the question is, what's the proportion of the offspring that are expected to be normal, that is, long wings and red eyes, and what proportion are expected to have dumpy wings and white eyes? Well, this question, it boils down, it can be condensed because the one parent, the big D, big R, can only produce one kind of gamete, big D and big R. Whereas the other parent can produce either a heterozygote, well, not heterozygote, it can be big D, little r, or little d, little r gametes. And so you can make that into a very simplified Punnett square where you only have one parent only has one kind of gamete, and the other parent can have either of two. So if you do the matching up after fertilization, big D, big R, little r, that would have red eyes and normal wings. Big D, big R, little r, again would have long wings because of the dominant allele at D, and red eyes because of the dominant allele at R. So the answer to the question is, what proportion of the offspring are expected to be normal? It would be 100%. And what proportion are dumpy and white? 0. Everybody follow that? Everybody see the logic behind it?
Yeah, I do.
Okay. What haven't we done yet? Okay, let's do number 7, phenylketonuria. It's a human autosomal genetic disorder. What does that mean? Autosomal genetic disorder. Anyone want to guess? We've talked about it. We know that there are 2 kinds of chromosomes. What are they?
X and Y.
Excuse me?
X and Y.
Those are not autosomes.
Autosomes.
Yeah, so what are autosome chromosomes?
They're diploid and haploid chromosomes.
Well, they can be, but
Or can
autosomes are in contrast to what?
The sex chromosomes,
Right.
Hmm.
So the autosomes are then what?
all the other ones.
All the other ones, right. Okay, so we know then that this gene for phenylketonuria is on an autosome. That means it's not on a sex chromosome. The affected individual cannot metabolize phenylalanine. So it's characterized by severe mental retardation if left untreated, and it's caused by homozygosity of the recessive mutant allele. So if 2 parents are heterozygous for the allele, what's the probability their child will have PKU? So again, we define the gametes in terms of what's diseased and what's healthy. Since we know that the disease is, it only shows up in the homozygous recessive condition. So that means that people with the disease are little d, and healthy can be big D, or big D, little d. And so the parents, it says that 2 parents are heterozygous for the allele. That means if we do the parental cross, is big D little d times big D little d. And our Punnett square shows the following. Big D, big D, big D little d in one square, and big D little d in another square. And finally, one square gets little d little d. So what does that tell us about, the question is, what is the probability the child will have PKU?
%25.
Right,
Yeah, 1/4.
25% chance.
And Professor, it doesn't matter to the girl or the boy, right? For the chance that they will have with this disease.
Right, and you've put your finger on an important point. The very first sentence here refers to autosomal. So that excludes any sex linkage. Doesn't matter if it's a boy or a girl. Okay, we haven't done earlobes yet. So in humans, the allele for free earlobes is dominant to the allele for attached earlobes. And a woman with attached earlobes has children with a man who has free earlobes. So what is the genotype of the woman? Well, the woman has attached earlobes, and the allele for free earlobes is dominant. So that means if she's got attached earlobes, what is her genotype? Attached is recessive.
Yeah, the small e's.
Right. So the woman is genotypically little f, if we have F for free. And the male then can be either big F, or heterozygous. So possibility one, if the male is big F times little f, all of the children would have free earlobes because they're all heterozygous. And possibility two is that if the male happens to be heterozygous, crossed with a homozygous female, then 50% of the F1s would be free. So out of 100 chances, 75 would be free and 25 would be attached. Any question about that? This one, this is pretty straightforward Mendelian inheritance. So we've done the back cross. We've done the Lesch-Nyhan syndrome and the bleeder's disease and the dihybrid cross and the monohybrid cross. So I hope that you all had a chance to go over these in advance of today, and that this presentation and discussion has clarified any questions you might have had. Because this type of question is going to come up as soon as Wednesday. We have a quiz, and the next test we have will also have Mendelian inheritance problems on it.
Professor, I
Uh-huh.
do have a question. So when is the next test will be?
The next test, hang on, I think I have it here. Will be the 26th of April. That's a Monday.
Oh, okay. Thank you.
Okay, anything else? Have any of you made progress on your cystic fibrosis project?
I've made contact with my group and we're doing some research, but we haven't gone over what we found yet.
Okay, have you divided up? Oh, that reminds me, something you need to do. I sent you an announcement indicating that you should make an appointment with me for a preview. And so far, none of you has, but did you all see the announcement?
Professor,
Yeah.
Kiyomi Anderson. I
Uh-huh.
tried reaching out to my group, but nobody answered. So I don't know if I'm like not contacting them the right way, but I haven't been able to get a hold of anyone.
Yeah, I didn't know how to contact people.
Say that again.
I didn't know how to contact people in my group either.
Email them.
Okay.
I did it through Canvas, but nobody answered. So I just
Okay, if you don't get a response soon, let me know. Send me their names. I will contact them. So we have to find out quickly if people choose not to be involved. And if they don't want to be involved, they won't get any credit for this project. But it also means that we may have to make adjustments to the groups. All right. Right now we have 10 groups. And if there aren't enough people to make 10 full groups, then we'll condense them down. But we need to know that soon because you have to be working on the planning and organization of the material.
okay.
Yeah.
Thank you.
Please, everybody, let me know if you're not getting a response from one or more of your group members. Anything else that anybody has on their mind?
Are you done with the light?
Excuse me?
I'm fine. Sorry.
Okay, that was not a question? All right, well, if there's nothing else, that's it for today. And on Wednesday, we'll start on a new topic and you will also have a quiz.
So we have a quiz for next, like in Wednesday, right?
Wednesday, yeah, the 14th.
Okay, thank you.
So is that okay to leave right now?
Yes, you can leave.
Oh.
That's all for today.
Okay, thank you.
Uh-huh.
So I was wondering how to email the people because they're not in here in the
You
class.
should be able to just put their names in the campus email
Okay.
and their address should come up and you can send them messages directly. If that doesn't work, well, it should work. But you can also get people's email addresses in WebAdvisor.
Okay. Thank you.
And if you have any trouble, just let me know and I'll try to help you out.
All right.
Okay, I'm going to stop sharing the screen. Anything else before we go?
Professor, what if what if they didn't respond? Do you said to email you?
Yes.
Okay. Okay, thank you.
Okay. Anything else? If not, I'm going to end the session. And I will see you all on Wednesday.